Hello everyone. Today we will discuss epidemiological study designs and topic. Um, we will cover these topics like introduction to epidemiological studies, cross-sectional studies, cohort studies, case control studies, interventional studies, comparative analysis, and study selection. We will cover all these topics. First, we explain their definitions, how to calculate them, what their strength, what their limitation, and their examples. First, introduction to epidemiological studies. What is epidemiology? Epidemiology is the study of the distribution and determinants of health-related states or events in specified population, and the application of this study to control health problems. Study design hierarchy. Observational studies, interventional studies. In observational studies, investigators observe without intervention. What is intervention? We will discuss later. And in observational studies, there are two types: descriptive, that the cross-sectional, analytical, cohort, case control. All these study types we will discuss in the coming topics. Interventional studies, investigators actively intervene. Experimental, randomized controlled trials, and we can say RCTs, quasi-experimental non-randomized trial. And the key concept that we will cover in the epidemiological studies is the exposure, the factor being studied, for example, smoking, medication, genetic variant, and you can add a lot of others related to your fields, and the outcome will be the health event of interest, for example, disease, death, and the bias. Systematic error in study design, conduct, or analysis. In a narrative and any meta-analysis or the review article, we can add a lot of studies. Their scenarios are different, their data are different, so to calculate their bias, that we can this is the complementary, this is the mandatory topic. If you write any publication, you have to calculate the bias that is presented between the studies. Confounding, when a third variable influences both exposure and outcome. Next is the cross-sectional studies. Cross-sectional studies assess exposure and outcome simultaneously at a single point in time or over a short period. They provide a snapshot of a population. Characteristics: time dimension, there should be a single time point. Direction, neither forward nor backward, like we can use it in the present time. Temporal sequence, cannot establish causality, also known as prevalence studies, surveys. Design structure, how we can follow these studies. If we will do population, then we will take a sample, then assess exposure and outcome simultaneously. Data collection method, which methods we can use to collect the data when we are doing the cross-sectional studies. Like we can do by surveys and questionnaires, physical examinations. It depends on the type of your studies and your field. Laboratory test, medical record reviews, imaging study. Measures of association. Prevalence ratio, that we can say PR, equal to prevalence in exposed divided by prevalence in unexposed. This is the formula. And a prevalence odds ratio equal to exposed with outcome and exposed without outcome divided by unexposed with outcome and unexposed without outcome. What are the strengths of cross-sectional studies? Number one, quick and inexpensive. Like we just say that in the characteristics, we only select one time point. So it's a quick and inexpensive, good for prevalence estimation, ideal for measuring disease burden, multiple outcomes, can study many diseases simultaneously. Like this is a cross-sectional, that's why we can study many diseases at one time. Hypothesis generation, useful for identifying association for further studies. Population representative, can provide generalizable data if properly sampled. Ethical, no intervention required. These are the strengths of cross-sectional studies. Next is the limitations. No causality, cannot determine temporal sequence, like chicken or egg problems. Survival bias, misses cases who died or recovered quickly. Recall bias relies on participant memory for past exposure. Incidence cannot calculate incidence rate. Reverse causality outcome may have influenced exposure. Poor for rare diseases. Need very large sample. These are the basic strengths and limitations studies. Next is the examples in health data science. That is, you can understand it in a better way. Example number 1, digital health adoption. Research question: what is the prevalence of electronic health record use among hospitals, and is it associated with hospital size? Design: what will be the design? We will do survey in this example. Survey all hospitals at one time point. And how to analyze: calculate prevalence of EHR adoption compared by hospital size. Example number 2, mental health and social media. What will be the research question? Is social media use time associated with depression symptoms? What will be the design? Online survey assessing current social media use and depression scores. What will be the limitation of this? Cannot tell if social media causes depression or depression increases social media use. This is the limitation and the critics of this. And the next is data analysis consideration. What we need to consider when we analyze data by the cross-sectional studies: use prevalence ratio or odds ratio, adjust for confounders using multivariable regression, logistic regression for binary outcomes. Consider survey weights if using complex sampling, account for clustering if applicable. Sample size calculation for comparing two proportions. This is the formula you can use, and these are the basic points just you have to calculate. The P1 and P2 is the expected proportion in each group, and the group which you are comparing to each other, like exposed and unexposed, these are the two control and the diseased groups. And the same way is the P equal to average proportion. Zeta alpha is critical value for significance level, zeta beta critical value for. What are the cohort studies? First, we will see the definition. Cohort studies follow a group of individuals who share a common characteristics or experience over time to compare outcomes between exposed and unexposed group. What are the types of cohort studies? Prospective. Cohort follow participants forward in time from present to future. Exposure assessed at baseline, outcomes occur during follow-up. Retrospective cohort, this is a historical. Use existing records to identify exposure in the past, follow forward to present to assess outcomes. Both exposure and outcome have already occurred. In the prospective cohort, we will follow the concurrent, like the time from present to future. And the retrospective cohort, we use the data from past to present. Ambidirectional cohort combines retrospective and prospective elements, uses historical data for initial period, then follows prospectively. What will be the design structure? First, the population, exposed group, follow-up, outcome, assessment. Unexposed group, follow-up, outcome, assessment. Measures of association incidence rate. IR equal to number of new cases divided by percent time at risk. Cumulative incidence. Risk equal to number of new cases, number at risk at start. Relative risk. RR equal to risk in exposed over risk in unexposed. These are the measures that we have to calculate when we will do studies. And the rate ratio equal to incidence rate in exposed and divided by incidence rate in unexposed. Attributable risk difference. AR equal to risk in exposed minus risk in unexposed. Attributable risk percent. We can also calculate the risk percent by have to have a large. Types of bias in cohort studies: selection bias, healthy worker effect loss to follow-up bias. Information bias, measurement error in exposure or outcome. Detection bias, if outcome assessment differs by exposure. Confounding requires careful assessment. These are the examples in health data science. For example number 1, Framingham Heart Study. What will be the design? Prospective cohort started in 1948. Population is 5,209 adults from Framingham. Follow-up ongoing for over 70 years. It's the long time. Outcome: cardiovascular disease, stroke, dementia. What is the impact? Identified major CVT risk factor like smoking, cholesterol, and hypertension. Example number 2, electronic health records cohort. Research question: does statin use reduce cardiovascular events? What will be the design? Retrospective cohort using EHR data. Exposed patient prescribed statins, unexposed. Matched patients not on statins, outcome: MI, myocardial infarction, stroke, cardiovascular death. Follow-up 5 years from first. Data analysis strategy: survival analysis. These are the Kaplan-Meier curves for survival estimation log. Rank test for comparing survival curves. Cox proportional hazards regression for adjusted analysis. Time varying exposure. Use time-dependent covariance in Cox models. Landmark analysis. Marginal structural models. Next is the case control studies. What are the case control studies? Case control studies compare individuals with a disease or outcome to those without, like the controls, and look backward in time to assess exposure history. Design structure. First, the population, cases with outcome, look back, assess past exposure. In the control without outcome, look back, assess past exposure. Control selection should represent exposure distribution of source population, must be eligible to become a case matching considerations. Control selection strategies. Number 1 is population control, hospital control, neighborhood control, friend relative controls. Random sample from general population representative but can be difficult to recruit. Patient with their other conditions from same hospital, convenient but may not represent source population. From same geographic area as cases helps control for environmental factors. Each and every strategy have different pros and cons. Friends relative control nominated by cases may overmatch on social factors. Matching, individual matching, frequency matching. In individual matching, each case matched to one or more controls match on potential confounders like age, sex, etc. and genders like this. Requires matched analysis, conditional logistic regression. Frequency matching controls selected to have similar distribution of matching variables can use standard logistic regression. These are the formula. You can measure the association of odds ratio, and for the rare disease, odds ratio is equal to RR, risk ratio. Exposure odds ratio, EOR equal exposed cases divided by exposed non-cases and divided by unexposed cases and divided by unexposed non-cases. Next is the strengths of case control studies: efficient for rare diseases, quick results, less expensive, multiple exposure, suitable for disease with long latency, practical. What are the limitations? Cases may remember exposure differently than controls, and the selection bias. Difficult to establish exposure preceded outcome. Cannot calculate incidence, no denominator of at-risk population, single outcome designed for one disease at a time, rare exposure inefficient if exposure is very rare, relative risk approximation, OR only approximates RR when outcome is rare. That's why it's good for the types of bias in case control studies. Recall bias, information bias. Cases may recall exposure more thoroughly than control, particularly problematic for subjective exposures. Mitigation, use objective measures, validate self-reports. Selection bias. Bergson's bias, hospital control may differ from population. Volunteer bias, cases more motivated to participate. Survival bias, cases who died are excluded. Mitigation, careful control selection, high participation rates. Now the observer bias. Like the bias is a thing that can create differentiation between the um, studies because they have different scenarios, different data. That's why we have to calculate the bias. Interviewers may prop case differently than control mitigation. Blind interview to case state. Now the examples in healthcare, health data science. Lung cancer and smoking. Case is patient with newly diagnosed lung cancer. In the control, patient without lung cancer from same hospital. Exposure smoking history obtained through questionnaires. Findings. Strong association like odds ratio equal to 10 to 20 for heavy smokers. Example number 2, medication safety study. Research question, is a new diabetes drug associated with heart failure? Design, what will be the design? Nested case control in EHR database. New heart failure diagnosis. Controls, what will be the controls in medication safety study? Matched on age, sex, diabetes duration. Exposure, new drug use versus standard therapy. Analysis, conditional logistic regression. Now we will discuss the interventional studies. What is the definition? Interventional studies involve active manipulation by the investigator to assign participants to different exposures or treatments and then follow them to observe outcomes. Hierarchy of interventional designs. Gold standard randomized control trials, we can say RCT, quasi-experimental, non-randomized interventional studies, randomized control trials, core principles, randomization. Random assignment eliminates, randomization can negate the, and you can say can cancel the biasness among the studies. So random assignment eliminates selection bias, balances known and unknown confounders, basis for statistical inference. Control group provides comparison to isolate treatment effect, types, placebo, active treatment, standard care, no treatment. Blinding. Single blind participant unaware of assignment, double blind participants and investigator unaware. Triple blind also include data analysts don't know what to do, and the purpose is that reduces performance and detection bias. Design structure, eligible population, randomization, then the treatment groups, then the follow-up outcome. In the control group, follow-up and outcome. Types of RCTs. Parallel group design. Most common design, participant randomized to one group for entire study, groups compared at end of the follow-up. Crossover studies. Participants receive both treatments in sequence, washout period between treatments. Each participant serves as on control. Requires stable condition, reversible treatment effects. Cluster randomized trial. Groups. Clusters randomized instead of individual, example schools and hospitals. Communities required when intervention is at group level, must account for intra-cluster correlation. What is a factorial design? Tests multiple interventions simultaneously. Each factor has 2+ levels, can assess interactions between interventions. Examples 2 by 2, 2 into 2 design tests, 2 drugs independently and combined. Adaptive designs allow modification based on interim data, can adjust sample size, allocation ratios, doses, maintains trial integrity through pre-specified rules. Non-inferiority trials test whether new treatment is not worse than standard, uses non-inferiority margin common for generic drugs, less invasive procedures. Equivalence trials test whether two treatments have similar efficacy, more stringent than non-inferiority. Randomization method, simple randomization, coin flip, random number generator, may result in imbalanced group. Small samples, block randomization. Ensure balanced allocation within blocks. Block size should be concealed. Examples, block of A, AAB, and you can arrange them in a different way. Block randomization. These randomization methods can reduce the biasness among the studies. Stratified randomization, separate randomization with strata ensures balance on important prognostic factors, for example, stratify by disease, severity, and site. Minimization. Adaptive randomization balances across multiple factors simultaneously, not truly random but highly effective. Response adaptive randomization, allocation probabilities change based on outcomes, more patients assigned to better performing arm, ethical but may reduce power, blinding strategy, placebo control, identical in appearance, taste, administration, essential for subjective outcomes, sham procedures used for surgical procedural intervention, ethical consideration important in the sham procedures, blinded outcome assessment, even if treatment cannot be blinded, assessors can be critical for subjective. What are the measures of effect? Absolute risk reduction, relative risk reduction, and the hazard ratio. These are the formulas that we use to calculate the data that we represented in RCTs. Phases of clinical trials. Phase number 1. First in human trials, small sample. Focus safety, dosage, pharmacokinetics, often in healthy volunteers. Phase number 2. Proof of concept. Sample will be increased like 100 to 300. Focus, what will be the focus in phase number 2? Like the efficacy, optimal dose, side effects in hospital with target conditions in the phase number 3. Confirmatory trials, large sample, we use the large samples, and the focus will be the efficacy versus standard treatment. Safety biases, basis for regulatory approval. Phase number 4, post-marketing surveillance, very large population, focus long-term effects, rare side effects, effectiveness. Intention to treat versus per protocol analysis. What is the intention to treat? Analyzes participants as randomized includes all randomized participants, preserves randomization benefits, more conservative estimate preferred for primary analysis. Per protocol analyzes only participants who completed treatment, may introduce bias, and can overestimate treatment effect, useful as sensitivity analysis. As treated, analysis based on treatment actually received, violates randomization, can be biased. What are the strengths of RCTs? Causality, strongest evidence for causal relationships, eliminates confounding, control bias, blinding reduces multiple biases, as we discussed the types of biases, precision, planned sample size ensures adequate power, regulatory standards required for drug device approval, protocol driven, standardized procedure enhance internal validity. In the limitations, expensive, high cost for infrastructure, monitoring personnel, time consuming, artificial setting, ethical constraints, cannot randomize harmful exposures, limited generalizability, strict eligibility criteria, created selected population, recruitment challenges, short follow-up, not feasible for rare outcomes, long latency diseases. Examples in health data science. Example number 1, Anglo-Scandinavian cardiac outcome trials. Intervention, amlodipine versus atenolol for hypertension. What will be the design? Double-blind, parallel group RCT, sample size in 19,257 patients. Primary outcomes will be non-fatal myocardial infarction and fatal CHD. Results, amlodipine superior. In the we can calculate the HR and p-value. Examples number 2, recovery trial, COVID-19 design, open label adaptive platform trial, intervention, dexamethasone, tocilizumab, hydrochloroquine, and others. Sample 44,000 plus patients. Outcome is 28-day mortality. Mortality impact, rapidly identified effective treatments during pandemic. Now we will discuss the quasi-experimental designs. When we will use these designs, randomization not feasible or ethical, natural experiments, policy evaluation, pragmatic settings. What are the types? Non-randomized controlled trials, before-after study, interrupted time series. In non-randomized controlled trials, investigators assign treatments non-randomly, may use alternation, convenience, preference, high risk of selection bias. Before-after study measures outcome before and after intervention. No control group, cannot rule out temporal trends, regression to mean. An interrupted time series, multiple measurements before or after intervention, examines changes in level and trend, control for pre-existing trends, strong quasi-experimental design. Difference in difference, compares change over time in intervention group versus control group, controls for secular trends and time invariant confounders common in policy evaluation. Regression discontinuity, design assignment based on cutoff score or continuous variable, compares outcome just above and below cutoff, strong design if assumptions met. Propensity score method uses observational data but attempts to mimic randomization, balances covariates between treatment groups, methods, matching, stratification, weighting, uh, covariate adjustment. Pragmatic trials. RCT is designed to evaluate effectiveness in real-world clinical practice. These are the pragmatic trials. What are the characteristics? Broad eligibility criteria, diverse setting, minimal exclusion, flexible protocols, outcomes relevant to patients and clinicians. Precise 2 framework rates trials on 9 domains from explanatory to pragmatic eligibility criteria, recruitment setting organization, flexibility, delivery and adherence follow-up, primary outcome, primary analysis. Data analysis for RCTs, primary analysis, intention to treat, pre-specified in protocol, typically using regression model. What are the survival analysis? Kaplan-Meier for time to event outcome, log rank text for comparison, Cox regression for adjusted analysis, handling missing data, multiple imputation, last observation carried forward, less preferred mixed models, use all available data. Subgroup analysis, pre-specified in protocol, test for interaction, adjust for multiple comparison, interpret cautiously. Interim analysis, pre-planned, looks at data, use alpha spending functions, independent data monitoring committee. Regulatory and ethical consideration, good clinical practice, international ethical and scientific quality standards protects rights, safety and well-being of participants. Institutional review board, ethics committee reviews and approves protocols, monitors ongoing studies, ensures informed consent. Data safety monitoring board, trial registration and reporting standards. These are all you have to do studies. At the end, the comparative analysis and the study selection. These are the analyses like cross-sectional, cohort, case control, and RCTs. And these are the features that we just compile in a tabular form, like the direction, time required, cost, casualty, rare disease, rare exposure, multiple outcomes, multiple exposure, bias susceptibility, and the major temporal sequence, sample size.